Hello, everyone. Thank you so much for joining us on this evening. We are very thankful to welcome Dr. Saver from UCLA. He is the primary investigator of the SWIFT Prime trial, which is one of our landmark trials that supports the use of mechanical thrombectomy in patients with acute ischemic stroke. My name is Arshleen. I am a fourth-year student at Jefferson, interested in interventional radiology and one of the chairs for our landmark trial series. This is under the umbrella of WCVIS, or the West Coast Vascular Interventional Society. The WCVIS is developing a training section to educate, recruit, and uh, include those passionate about pursuing 100% endovascular and interventional surgical career with a comprehensive and longitudinal clinical care, which is why this series is so important because it helps us make clinical decisions. The direction of VIR as a specialty will be defined by those who don't compromise in providing the best possible care to their patients. And so we have many sections from the pre-medical students to medical students, residents, and early career trainees in VIR. And we have a lot of ongoing projects for those interested and who would like to get involved. We have several series in addition to our landmark trial series. We have our match series, acute care series, how to crush your sub-I, neurointervention, and our transnational vascular interventional journal club, who is having a session later on this week, actually, for those that would like to join. We have excellent mentorship and residents and attendings who are willing to mentor students and speak to them. And this is a big reason for why I ended up applying VIR to begin with. So I highly recommend anyone to get involved if you think you're interested. If you'd like to stay updated, we have a QR code that you can scan, and feel free to follow us on social media. We push out a lot of our events and updates on there, and have all of our sign up links. Finally, to introduce our guest, we have Dr. Jeffrey Saver. He is a distinguished professor of neurology at UCLA. He is the Carol and James Collins Endowed Chair and Vice Chair for Clinical Research. He is the director of the UCLA Comprehensive Stroke and Vascular Neurology Program. And most of all, he is the primary investigator of the SWIFT Prime trial. Dr. Saver is the expert, so I won't belabor it too much, but as a brief overview, the SWIFT Prime trial is one of our landmark trials along with Mr. Clean, and there's 5 major trials that supports the use of mechanical thrombectomy in patients with anterior circulation large vessel occlusion. And this trial compared the solitaire retriever with intravenous TPA against intravenous TPA alone. And the result ended up showing a significant decrease of in the modified Rankin scale between 0 and 2 compared with IV TPA alone. And so, without further ado, I'm going to let Dr. Saver take the stage and tell us about his experience designing this trial, any challenges he faced, and his learnings, and just what his experience was like. Thank you, Dr. Saver. Really appreciate your time.
Well, thank you. What a wonderful group you all have. It's a pleasure to be here. And I thought I would tell some of the behind the scenes stories from the SWIFT Prime trial and the studies that preceded it. I have a few anecdotes that I'll go through. I think it's helpful for my own experience to know I had the pleasure of coming to UCLA where the first retriever device was invented, the coil retriever, the Mercy retriever here by a French neuroradiologist, Pierre Gobin. It was a kind of corkscrew that you twist into the clot and pull it out like pulling a cork out of a wine bottle, not an accident. That was a French interventionalist who invented it. And so here at UCLA, we were part of the very first human trials of these devices. And the Mercy device was approved in 2004 based on a non-randomized study. as a tool to open clots. Didn't say it made patients better. Very controversial at the time. Um, and a year after it was approved at a satellite meeting, giving it a talk, and I was debating Kira Becker, who was one of the panel members when the Mercy device was approved, and she had voted against its approval. And we were debating whether the Mercy device was beneficial. And I went first, and I came out and I said, I agree there's not definitive evidence right now, but I think when we complete the trials, and we have started them, it is going to be shown beneficial. And I'm so certain it is. I pulled out $100, put it on the lectern, and said, I will bet you, Dr. Becker, $100 tonight that when we're done, this will be shown beneficial. And it was very unfair of me, but Kara, to her great credit, came out and opened by saying, Dr. Saver, I will take that bet. And then over the next 5 or 6 years, our group led the MR rescue trial funded by NIH, and there was also the IMS3 trial of that generation's the stent retrievers, coil retrievers, and IA lysis. And they were both reported neutral in 2012 at a meeting in Hawaii. And when I came off the lectern in Hawaii, I went up to Dr. Becker and I gave her $100. And I really had thought it was going to work, but it was a very difficult device to use. It only clearly got the artery open well 25% of the time. And if you don't open the artery, it's not going to make patients better. Fortunately, just as we finished the trial, this often happens in the device world as you're developing therapies, the technology advances in a way that makes what you just showed completely outmoded. So the stent retrievers arrived just after we finished these trials. And I was first involved in the SWIFT trial, which compared the stent retriever to the coil retrievers and showed that they work better. And that set up the SWIFT Prime trial and the other trials of the time. SWIFT Prime was distinctive in a few ways. Most importantly, it was the only trial that was done under FDA overview and being done to support a label change in the US. There were international trials and trials not confined to a single device. This was the only one that was going to lead to a label change. And as we did the trial, a couple of snapshots. Mayank Goyal, who some of you probably know from Calgary, rapidly emerged as the most fiery and dynamic of the interventionalists in the trial, and became kind of a co-PI with me. And we each complimented each other well. I think as a non-invasive neurologist, was good at bringing all the different groups in the trial together, and I had time to think about what to do. I didn't have to be in the cath lab all the time, which is an advantage. And but Mike was someone who would go out and energize the troops and all over the world, getting people to enroll and enroll quickly. There were two books at the time. One was about Abraham Lincoln, Team of Rivals, about how he brought his cabinet together of all these different people. And Mike said, that was my role. And the other was about Steve Jobs and how when he entered a room, reality was warped. And that's what I thought Mike's effect was when he'd speak to people about the trial. So as we were going along, there were several trials underway. The first that was going to be reported was the Mr. Clean trial. And in the US, we didn't know much about the Mr. Clean trial. What I had heard, it turned out not to be the case. But what I'd heard, and I think what most people in the US by secondhand incorrectly understood, was that they were using old devices and that we should not get our hopes up. That was somewhat unlikely the trial would succeed, but we shouldn't take that to mean that our trials would succeed. And uh, then I was asked to be one of the reviewers by the New England Journal of Medicine for the Mr. Clean submission. And I accepted. I thought, okay, now I'm going to find that this is disappointing and figure out how to position Swift Prime around it. And I was in my van pool going to UCLA, surrounded by dental hygienists and uh, janitors, when I was reading their article, and I realized the world changed. I looked down and I looked up, and it was a whole new era. And but I couldn't tell anyone for 3 months. Um, and then uh, when Mr. King was reported positive at the World Stroke Meeting in Istanbul, got a standing ovation there. All of the other trials that were active had to decide, were we going to continue enrolling, given this highly positive trial, or stop and take a look at our data in an unscheduled way to see whether it was ethical to continue. And so for Swift Prime, we had a meeting of the steering committee. There were 6 members on the steering committee. The sponsor wanted Medtronic, the predecessor Medtronic, wanted to continue the trial because they wanted definitive data. They wanted to go to the full enrollment numbers and have definitive data for FDA. But of course, it was very compelling data from Mr. Clean that we should potentially not do trial. So as chair of the committee, we discussed it. We had a vote, and they turned to me to see what I would say first. And I said, nope, I'm chair, I'll go last. I didn't want to influence people. Plus, I figured if I said something, people might come along with me, and I didn't want it to really get a democratic opinion. And I also thought if I went last, most likely people would have voted one way or another, and my vote won't matter, and nobody could blame me. And they went around the room, and the vote came in 3-2 for stopping. And then I had the 6th vote, and if it was a tie, it probably would have gone with me breaking the tie. But I said we should stop and take a look, and that's what we did. And then a month later, I'm sitting in my office, and I get a call from Wade Smith, who was the chair of the DSMB, neurointensivist at UCSF. And he started off by saying, Jeff, this is one of those calls that you wait all your career for. And then he told me the results of the Swift Prime trial that were highly positive. So that was terrific. And the last anecdotal snapshot I'll tell you is about the presentation of the results at the International Stroke Conference in February. 3 of the trials were being presented, Revascat and Escape and Swift Prime, in the same plenary session. And Swift Prime was slotted last. So I was very disappointed because we would just be the clear one. And yes, we were distinctively the one that would lead to a new American label, but key results would be very similar. But unexpectedly, it was one of those times when there's this periodic concern about p-values and overuse of you can use p-values without concern for the primary endpoint. They are confirmatory studies. You have a hypothesis, you're seeking to confirm it. That's what a p-value is for. But the because of this general uh, hesitation, both of the other presentations only presented confidence intervals. Neither presented p-values. And the other thing that had happened was over the prior 20 years, there had been the TPA controversy and TPA war with emergency medicine, where inappropriately emergency physicians criticized the thrombolytic trials. And a main claim of theirs was they're too small and the p-values, although significant, are fragile and not reliable. So when I presented the results, I had on the slide the p-values, and I purposely drew it out for drama and said this was positive with a p-value of 0.0001. And the audience erupted in applause. Rosemary Robertson, who is the scientific director of the American Heart Association, a cardiologist, came up to me afterwards and said it's the first time she's ever heard applause for p-value. And the and then then it was off to the races. The trial did lead to the change in the FDA label, and and led to the new era of highly effective reperfusion therapy we have now. So that's kind of multiple snapshots along the way. Let me stop there, see if there are particular other things you'd like to ask me about.
That was amazing. Thank you so much. It was great to hear a lot of these anecdotes. And something that I was wondering, especially as medical students and early trainees, when we are starting our research projects, and especially ones that are kind of perspective based, one of the biggest hurdles is recruitment. And you said that your job in Swift Prime Trial, a lot of it in the beginning is gathering your participants and encouraging people to enroll patients. And in a pathology like stroke and an intervention and in an active intervention in the cath lab that's new, I can imagine that sometimes there might be some hesitation to enroll. I was kind of wondering your approach to gaining more enrollment into your trial and how you spoke to all these different physicians and kind of your experience with getting adequate enrollment.
Yeah, that is always challenging in a procedural trial where patients might go through procedure or not, and everyone tends to personally think the procedure is going to help. Otherwise, you wouldn't be doing the trial, even though personal equipoise is not the basis for randomization. It's scientific equipoise. So for Swift Prime, uh, we had the background that the MR Rescue and IMS3 trials of intervention had failed. And those trials failed in part because people were unwilling to enroll all patients, and they preferentially cherry picked or reverse cherry picked patients. They treated patients most likely to respond outside of the trial, cherry picking, leaving in the trial patients least likely to respond, reverse cherry picking. And uh, when those trials came in neutral, the payers, the insurers in the US, began to issue policies that they would no longer cover thrombectomy. And uh, that helped us a lot. That was a very galvanizing influence. First, the interventionalists, I think everybody realized that given that neutral trials had come in, everyone needed positive data for the field to really flourish um, and to really change systems of care, we needed an anchor of positive trials. Plus, the fact that soon patients might not be reimbursed. It wasn't quite the point that they could only be reimbursed if they were enrolled in the trial. It hadn't reached that stage, but that was beginning to loom. Helped in the US to motivate people. Now, it was still a challenge, and in Europe, the European trials had a different experience. Um, the But I think and our experience in Europe was the same, but maybe a little bit more cherry picking there. And in the US, there was still cherry picking going on. And, I can say when I had a 45-year-old come in with left M1 occlusion and uh, hemiplegia, aphasia, and clearly quality of life threatening ending occlusion in place, uh, being able to randomize that patient is very challenging to do. Another aspect is you have to randomize for the acute interventions within a space of minutes to get patients, so you don't have time for an extended discussion with the patient. And we had talked about potentially doing the trial under the exception from informed consent, EFIC policies that um, recognize that in these hyper urgent treatments, you can't really have a good consent conversation. The only way we're going to get data is to do the trial under uh, deferred consent, but to make sure it's ethical, to first do community consultation and public disclosure before you start the trial. But there's a lot of effort required in that public disclosure and community consultation, so it slows down the start of the trial. Uh, so that helped us a lot. We had, league tables every month. We'd report how each site was enrolling. We had the sites that had succeeded in enrolling give presentations at the meetings to the sites that were having trouble. We had mentors from high enrolling sites speak to the sites that were not enrolling. Sites that were really not enrolling were put on probation, threatened with being removed from the trial. That's the stick. On the carrot side, we said that the top maybe 5 trials at 5 sites in the US and 5 in Europe were 3 and 3 that had the highest enrollment. Their investigators would be top line authors on the paper. Um, so those were some of the things that we did to encourage enrollment.
Absolutely. That's great insight. I know like we were talking about coil retrievers earlier and how they weren't necessarily seen as effective. Given that history, when you were kind of pitching this trial or this concept to people in the field, both on the procedural side, on the neurology side, did you get any pushback when you brought up this the solitaire and kind of talking about how it might be better than the coil retriever? Were people wary at all, or were people kind of open to the idea?
Uh, 2 head-to-head randomized trials had been done of the stent retrievers against the coil retrievers. The SWIFT trial tested the solitaire device, and the Trevo trial tested the Trevo device, and both came in showing much better technical success uh, and similar or better safety with the new devices. So with those trials in hand, there was no difficulty telling people uh, this is a uh, completely different uh, uh, technology, a much more successful technology, a much greater chance of success with the trial. And, Swift Prime was an industry trial. The trials that we performed were a mix of academic trials, of mixed industry academic trials, and of industry trials. Uh, so the uh, the original the prior trials, MRSQ and IMS3 had been done as NIH funded trials. They were pure academic trials. And I think that reflects the sense that there weren't big industry players developing these devices at the time because they hadn't been shown beneficial. There was not yet a market really for them that was of any size. And, uh, and so you had to do things for the public good. So we got NIH funding. But once these devices came along that were much more effective, industry saw a chance to really have market makers and put the money in. And I'll tell you, here's a secondhand story about how Medtronic or its predecessor uh, decided to develop the stent retriever because Solitaire was a releasable stent. You could go up, and if you wanted to, there was an electrostatic connection. You could leave the stent in place, and it was thought that that might be something that we ended up doing a lot. And it wasn't developed for retrieval. It was developed to treat different types of intracranial arteriopathy. Uh, and But one day, the UMass lab, Ms. Gunis, and others have run the most productive preclinical technology lab for endovascular devices for the past 25 years at UMass. And the uh, folks from the company were visiting the lab, and the lab was doing experiment in a large animal, and by accident had a clot go to an artery where it wasn't supposed to be. And they tried to take it out with the hooks or other things that were available at the time, and they couldn't. Then they decided to go up with the Solitaire device, and they went up, and boom, boom, it was out. And I think the company representatives turned to each other and said, they went back to headquarters and said, "We've got a new idea for this device." And that's how it came about.
That's amazing. These are the kinds of things that we don't hear when we read the paper, but that's awesome. Um, and then I guess from your point of view, if you were to lead another trial like this again, are there any learnings that you know now that you didn't know earlier that you think others should know who are looking to get into the research space, or is there anything you maybe would have done differently in hindsight?
I don't think so. It was a trial that was well matched to its time. It's hard to replicate. We are doing, as your journal club is going to be addressing these DMVL trials and low NIH stroke scale trials and these indication expansion trials, trying to take thrombectomy into other patient populations. And it's a lot harder now because we have with a very positive fundamental result known. It's hard to go to adjacent populations and get people to randomize. And it's also unlikely that there's going to be a cutoff of funding because it works so well. Um, so, uh, so that's a challenge. Yeah, I will say one aspect of these original trials, and there were originally 5, and then 2 more late trials from France and Great Britain came in. So there were 7 trials of that wave of small to medium core early treated patients. And we joined together to form the Hermes collaboration, pooling individual patient data, and uh, and started looking at that data set for a variety of other lessons to be gleaned besides the primary top line results. And now there have been about 40 papers from the Hermes collaboration. And that is one way for junior people to really begin to make progress, because you can't go from 0 to 60, come out of fellowship and be the PI of a 30-center international trial. But the stepping stones to get there, there are lots of other roles on clinical trials that can be filled that give you experience and also make you known to the leaders who are going to be selecting the future leaders of the trials. One is to propose secondary analyses, and most of the trials will have a time period a year after the trial is done when they are open for new proposals to come in. They preferentially will have people who did the trial be able to analyze interesting questions, but if you come in with an interesting question that no one else is analyzing, generally they're very happy to look at it. It's another publication for everybody. So looking for that. There are other roles in these trials, serving on different committees. There's clinical events committees to determine if a complication was related to a device, or was a second stroke or not. There's data safety and monitoring boards to look at the aggregate data and determine if it's safe to continue. There are internal recruitment committees where try and get people to focus on ways to go forward. There are committees about outreach to different communities to try and get a breadth of participation. So there's a variety of these supporting leadership positions that exist in the clinical trial world that one can do, and that then can gradually give you the education and experience to be able to get to the lead. It's also good to start your own trials, but the studies that people can do early in the career on their own, are going to be single center, much more limited in scope. But there are interesting things that one can do with early phase 2 studies, looking at feasibility and safety endpoints, and again, mounting a trial and completing it with you as the leader, even if it's a single center trial, is always an impressive feat that will get people's attention.
Absolutely. That's great advice for a lot of us that are interested. Michelle, looks like you have a question. Would you like to go ahead?
Hi, Dr. Saver. Thank you for taking the time to come and speak with us about such an exciting time and such an exciting finding for stroke management. My first question for you is, how did you manage, I guess, or maybe if you had the confidence, you didn't have the feelings I did, but how do you manage the hesitancy that maybe something novel could be not what you had hoped? Because I remember I used to be a clinical research coordinator for pediatric stroke trials. And I remember enrolling the very first patient for a steroid for vasculitis stroke. And we did it in a patient before we knew they had vasculitis or not. And I remember being so scared to enroll that patient because it was a gamble. We were trying to treat them prior to knowing if they had vasculitis. And so I was always so nervous to approach patients and try to recruit them prior to knowing. But of course, like as we see retrospectively, it was very beneficial and it was such a huge success. So how did you navigate those emotions in the moment, or did you even have those emotions in the moment?
Well, I think to get into the trial, you needed imaging, CTA or MRA confirmation of the target occlusion. And that's one way in which this trial differed from IMS-3, not from our rescue, but IMS-3 was early CTA days. It wasn't available everywhere. So some patients were enrolled without the target lesion. So we knew what the patient had. And in this trial, the issue was for this device we think works, are you going to be able to live with yourself with the patient being assigned to the medical group and having a bad outcome? Not so much that the device would behave badly. We already knew its profile pretty well. But a related note, I had been the PI of the NIH FastMag trial, magnesium sulfate, in the ambulance as a neuroprotective drug that we did for 8 years in LA and Orange County. We enrolled 1,700 patients in 300 ambulances at 60 hospitals. The time from last known well to start of magnesium or placebo was 45 minutes. Which was the goal, to show that you could get drugs started faster if you went into the ambulance. And that was an NIH-funded trial. And when you do an NIH-funded trial, you're doing everything. You're doing the designing the CRFs, you're running the data archive, you're arranging for the monitoring. It is a huge undertaking. When you do an industry trial, the industry is doing the running of the trial. You're just doing the design and strategic top-level thinking and interpretation. And the industry is pretty good at running a trial, so it's a lot easier. But after pouring 10 years of my life into magnesium and FastMag, we got the results in early 2014 that it was neutral. That was a disappointing result with the agent we were testing. Now, we had designed the trial that we were testing this mechanism of giving the drug in the ambulance, as well as testing the drug itself. So it couldn't be a total loss if we succeeded in showing pre-hospital delivery was attainable. So it wasn't a home run, it was a double. But still, that was very disappointing. And but fortunately for my psyche, before I had a chance to wallow in that disappointment for too long, 6 months later, the SWIFT prime result came in highly positive. And so that helped a lot. And, stroke has been a very challenging area to do clinical trials. There's already a very high failure rate across all of medicine for new interventions, but especially in brain and its complexity. And I have led or participated in over 150 trials. Of those, I think 6 were clean win positive trials, and the rest were not. But it's the case that one positive trial makes up for 100 negative trials. Being persistent eventually paid off.
Absolutely.
Yeah, thank you so much for that insight. My second question was with the advent of tenecteplase and these trials saying that tenecteplase is as efficacious as TPA and almost even superior to TPA. Do you see a trial upcoming, or is there a trial ongoing with thrombectomy and tenecteplase?
There have been a few trials already of thrombectomy and tenecteplase. Go back to the Australian trial from several years ago and recent trials out of China. And it, as in other trials versus TPA, tenecteplase is as good or a little better, but not dramatically different. There are trials of novel approaches to thrombolysis. that I think are promising. The NIH sister trial is looking at drugs that affect von Willebrand's factor and can potentiate the effects of lytics. There are other novel thrombolytics industry is developing that claim that they don't have hemorrhagic side effects. There's the trials of giving lytics intraarterially after successful thrombectomy to clean up distal thrombi. So the space of combined therapy is an active one, both in trying to get better agents and better use of our current agents.
Thank you. That was great questions, Michelle. I think another question that I had, especially a lot of the research or the landmark trials that we see in IR, a lot of them are industry sponsored. And I was kind of wondering, with your experience on the academic side of things, and of course, we all try to, conduct unbiased research, is kind of wondering what the interaction is like with industry and these industry sponsored trials who are making these devices a lot of the times, and your experience with creating this trial design and interacting with the industry sponsor and what that relationship was like throughout the trial.
Sure. There's a wide range of experience. The big device companies, Boston Scientific, Abbott, Medtronic, have a very sophisticated clinical trials group, regulatory group, and they do trials very well. And if you look at studies of trial quality in the literature, industry trials have regularly had higher quality than academic trials that have been reported. On the other hand, the smaller device companies often have people who haven't had trial experience and don't understand that they can't look at blinded data or other aspects. And there are and the range of relationship between the academics and the industry sponsor is across the spectrum. On the one end, there are arrangements like with the TIMI group in cardiology, where you have an academic consortium that does multiple trials, and the sponsor comes to them and pays them a lot of money to do the trial. But the academic consortium is in substantially, they make sure they keep getting along with the sponsor, but they have much complete autonomy in making final trial decisions. Then on the far other end, there are trials in which the company's making all the decisions and academics are there, but they're really just figureheads. And that should never happen, and one should never allow oneself to be involved in such a trial, but it does happen. Most trials are in between, where there is somewhat a collaborative relationship between the sponsor and the steering committee. The sponsor retains final authority over some aspects, but the steering committee also retains authority over some aspects. One example I'll give is of the PRISMS trial of TPA for non-disabling stroke. That was a drug trial, Genentech, and when the trial was about 60% enrolled, enrollment had been slow, and Genentech leadership decided to stop the trial because it was taking so long. And they wanted the steering committee to agree and issue a notice that they were stopping the trial. And the steering committee thought, we haven't answered the question. This is still important in clinical practice. We want the trial to continue. And we didn't have the, Genentech spending their money. We can't make them continue. But we held, I actually, I was on the steering committee, and as a point of order, I called for a vote from the academic steering committee about whether they thought we should stop the trial or not. And it was overwhelming that the steering committee voted against stopping the trial. So you can take stands like that. Now we continued getting along with Genentech. The trial came in negative, clearly no benefit. So they didn't know that, but it turned out to be the right decision for them. And we continued, collaboratively reporting the results. And I think they understood where we were coming from. But another aspect is a fundamental obligation of a clinical trialist is to complete the trial. Trials run into all sorts of unexpected difficulties. They run for 5 or 6 years. They're asking a single question. That question might become outdated. The world changes around them while they are stuck in the moment when that question was framed. And I just forgot the brilliant point I was going to make with this, but oh yes, so when you're a trialist, you have to be ready to compromise and adjust on some aspects. And because a fundamental obligation of the trialist is to complete the trial. If you walk away from the trial and it falls apart, that's not helping anybody. So you can't be too rigid, and this has happened to some investigators, and they blow up and walk away, and they get to be purists, but they're not getting much done. On the other hand, you have to have a lines that you won't cross and say you won't go there, and say you can kick me out because I can't support that. So you have to walk that line.
Absolutely. What do you think are some of those areas where you can afford to compromise, or without compromising the integrity of the trial, or like what are some like practical compromise that you think should be made?
One is on entry criteria. Frequently you start off with pretty narrow enrollment criteria because you're looking for the patients who are going to be perfect for informing you about the therapy. And but it turns out those patients are few and far between, and enrollment is behind. So you have to be ready to compromise on the enrollment criteria to let in patients who may be somewhat less responsive to therapy, but that are much more common on the ground in order to get results. You may have secondary endpoints in the trial that are of great scientific interest. When we did the RESPECT trial of PFO closure for preventing recurrent stroke, we started off with a migraine secondary endpoint because of the potential that PFO closure helps migraine as well. But 100 patients in, the company said, filling out these migraine questionnaires is taking a lot of time, and that's not the point of the trial, and we we want to stop it. And we said, we understand, that's okay. That's not the core aspect of the trial. And frequently you have to sacrifice some of your cherished secondary endpoints in order to make the trial doable.
That makes a lot of sense. Thank you. We are approaching the end of the hour, and I do want to be respectful of everyone's time as we close. Does anyone have any other questions for Dr. Saber? Any questions? If not, we do really want to thank you, Dr. Saver. This was great insight and something that, we read these trials and we learn about how they shape the landscape of clinical decision making, but rarely do we learn the thought and the effort and the little stories that you told us that go behind all of this and behind the scenes. And it truly is inspiring and makes the field of research and creating the basis for good clinical decision making for patients all the more rewarding. So thank you so much, and I really appreciate your time. We know you're very busy.
Thank you so much. And I look forward 10 to 15 years from now to going to the meetings and hearing your trials being presented.